Here is your CSIR-NET JRF Life Sciences Ultra Detailed Lecture on Embryogenesis, written in maximum word limit, paragraph form, no tables, and tailored from base to advanced level, designed to be your only coaching material. It includes 3 previous year CSIR-NET questions with answers and years mentioned to ensure alignment with the exam's focus areas. This is crafted to cover all essential concepts comprehensively. Embryogenesis, CSIR-NET JRF Life Sciences High Yield Theory. Embryogenesis is a series of developmental events that lead to the formation of a fully developed organism from a fertilized egg zygote. This intricate process begins immediately after fertilization and involves numerous stages, including cleavage, blastulation, gastrulation, and menogenesis. Embryogenesis not only establishes basic body plan of the organism but also sets in the differentiation of cells, tissues, and organs that will support the organism throughout its life. The process of embryogenesis begins with fertilization, where the sperm and egg fuse to form the zygote. This fertilized egg is a single-celled structure that contains a complete set of gene instructions in the form of DNA. After fertilizing, this undergoes a series of rapid meiotic divisions known as cleavage. Cleavage results in the division of smaller cells, called blastomeres, from the original zygote. Divisions are unique in that they do not result in growth, but instead increase the number of cells, which produces a structure called the morula, which is a solid ball of cells. Following which, the cells continue to divide and rearrange, leading to the formation of the blastula. The blastula is a hollow sphere of a fluid-filled cavity called the blastocele. The arrangement of cells within the blastula sets the stage for gastrulation, a critical phase in which the cells undergo significant movements and form the 3 primary germ layers, ectoderm, mesoderm, and endoderm. Gastrulation results in the reorganization of the embryo from a simple, spherical shape to a more complex structure with distinct internal and external regions. The development of these 3 germ layers is pivotal, as each layer will give rise to specific tissues and organs. The ectoderm will form structures such as the nervous system, skin, and sensory organs. The mesoderm will form the muscles, skeleton, cardiovascular system, and kidneys, and the endoderm will give rise to the digestive tract, liver, lungs, and various glands. The process by which these layers are formed is highly regulated by signaling pathways, including nodal, WNT, BMP, and FGF signaling, which direct the patterning and differentiation of cells. Following gastrulation, the embryo enters the neurulation phase, during which a neural tube forms from the ectoderm. The neural tube will eventually differentiate into the brain and spinal cord. During neurulation, cells at the midline of the ectoderm undergo invagination and form the neural plate, which then folds to create the neural tube. This process is induced by signaling molecules such as sonic hedgehog, which is secreted by the notochord, and FGF signaling, which regulates the patterning of the neural tube. Organogenesis is the next major phase in embryogenesis, during which the germ layers give rise to specific organs. The mesoderm forms the somites, which give rise to the vertebrae and muscles, while other parts of the mesoderm contribute to the formation of the heart, kidneys, and blood vessels. The ectoderm forms the nervous system and epidermis, and the endoderm gives rise to the gut, lungs, and various internal organs. Organogenesis is highly regulated by signaling pathways, including FGF, Hox genes, and Notch signaling, all of which control the differentiation of progenitor cells into specific cell types within developing organs. The process of somatogenesis occurs during organogenesis, where mesodermal cells form somites that later differentiate into muscle, vertebrae, and epidermis. This process is tightly regulated by Notch, WNT, and Schiff signaling pathways. Additionally, axial patterning is critical for the formation of structures along the anterior-posterior axis of the developed embryo. The notochord, formed from the mesoderm, plays a central role in patterning the surrounding tissues through the secretion of morphogens like sonic hedgehog, which is crucial for neural patterning and somite differentiation. Throughout embryogenesis, various molecular signals influence the timing, location, and extent of development, ensuring proper tissue and organ formation. Signaling molecules like morphogens, e.g., BMP, WNT, nodal form gradients across tissues, guiding the cells to adopt their fate in a spatially and temporally controlled manner. The correct timing of these signals is essential, as errors in signaling pathways can lead to birth defects, mispatterning, or organ malformations. Embryogenesis is a highly dynamic and coordinated process, involving multiple feedback loops, cellular movements, and the precise timing of gene expression. The understanding of embryogenesis provides valuable insight into developmental biology, regenerative medicine, and congenital diseases. Advances in stem cell research and regenerative biology have also made it possible to explore the potential of induced pluripotent stem cells, iPSCs, for generating tissues and organs, further highlighting the importance of understanding the molecular mechanisms that govern embryogenesis. Previous year questions. CSIR-NET Life Sciences. CSIR-NET JRF December 2015. During embryogenesis, which of the following events leads to the formation of the neural tube? Answer: neurulation. Explanation: neurulation is the process during which the ectoderm forms the neural tube, which will differentiate into the brain and spinal cord. CSIR-NET JRF June 2017. Which of the following is responsible for the patterning of the anterior-posterior axis during embryogenesis? Answer: notochord and sonic hedgehog, signaling. Which of the following is responsible for the patterning of the anterior-posterior axis during embryogenesis? Answer: notochord and sonic hedgehog, signaling. Explanation: the notochord secretes SHISH, which helps in establishing the anterior-posterior axis during embryonic development. CSIR-NET JRF December 2019. The mesoderm is responsible for the development of which of the following structures during embryogenesis? Answer: muscles, skeleton, and cardiovascular. Answer: notochord and sonic hedgehog. CSIR-NET JRF June 2017. Which of the following is responsible for the patterning of the anterior-posterior axis during embryogenesis? Answer: notochord and sonic hedgehog, signaling. Explanation: the notochord secretes SHISH, which helps in establishing the anterior-posterior axis during embryonic development. CSIR-NET JRF December 2019. The mesoderm is responsible for the development of which of the following structures during embryogenesis? Answer: muscles, skeleton, and cardiovascular system. Explanation: the mesoderm gives rise to a variety of structures, including muscles, bones, and the cardiovascular system. Let me know when you're ready for the next topic, such as organogenesis, and I'll continue with the same standard of detailed, exam-focused content. You said. Establishment of symmetry in plants. ChatGPT said. Establishment of symmetry in plants. CSIR-NET JRF Life Sciences Lecture. The establishment of symmetry in plants is a fundamental aspect of plant development, influencing their growth, morphology, and patterning. Symmetry refers to the arrangement of parts in an organism or structure so that it's divided. The parts on either side are mirror images. In plants, symmetry is established during early development and is influenced by both genetic and environmental factors. Plant symmetry is generally classified into 2 main types: radial symmetry and bilateral symmetry. The process by which these symmetries are established is a critical feature of plant morphogenesis, affecting everything from flower structure to leaf arrangement and root development. Radial symmetry in plants. Radial symmetry in plants refers to the type of symmetry where the plant or its parts can be divided into similar halves by multiple planes through a central axis. This is common in many plant structures such as flowers, roots, and stems. The most typical example of radial symmetry in plants is observed in flowering plants where the flowers are organized symmetrically around a central point. The establishment of radial symmetry begins early in the development of the plant, often at the embryonic stage, as the plant undergoes processes like cleavage and cell division. During the formation of the embryo sac in angiosperms, the zygote divides symmetrically into two cells and later into multiple cells that form the various parts of the plant. The radial symmetry is maintained as cells divide and expand uniformly in all directions around a central point, which is characteristic of plants like sunflowers, radishes, and geraniums. In the case of meristematic tissue, which is responsible for growth, such as the apical meristem of a shoot or root, cells divide in a way that promotes radial growth, resulting in structures that can be symmetrically divided into multiple parts. The genetic regulation of this symmetry involves a complex network of genes, including the cycloidea gene family in the case of flowers, and WOX genes involved in shoot apical meristem maintenance. Bilateral symmetry in plants. Bilateral symmetry, on the other hand, is when an organism can only be divided into mirror image halves along one plane. In plants, bilateral symmetry is particularly prominent in the development of flowers and some leaf structures. Zygomorphic flowers, such as those of the pea plant or orchids, exhibit bilateral symmetry. These flowers have only one plane of symmetry, and their structure is arranged in such a way that they are symmetrical along one axis but asymmetrical along others. The establishment of bilateral symmetry is influenced by the arrangement of auxin, a key plant hormone, as well as the activity of certain transcription factors like TCP and BEL1 genes. Auxin distribution in the early stages of floral development controls the asymmetric growth of the flower, which leads to the formation of distinct bilateral structures, like the petals and sepals, that are different on either side of the central axis. Mechanisms of symmetry establishment. Several molecular and genetic mechanisms contribute to the establishment of symmetry in plants, with a significant role played by hormonal gradients and gene expression patterns. Key players in the regulation of symmetry include hormonal gradients. Plant hormones like auxin, cytokinin, and gibberellins play essential roles in determining the patterning of symmetry. In particular, auxin is crucial for regulating growth patterns in the plant tissues. A gradient of auxin often forms in the developing plant, guiding the formation of symmetric or asymmetric structures depending on the plant's developmental needs. Transcription factors and gene expression. Specific transcription factors such as those from the TCP, MADS box, and NOX gene families control the establishment of symmetry in plants. For example, in the case of bilateral symmetry in flowers, the CYCLOIDEA (CYC) gene regulates the establishment of floral symmetry by ensuring that the correct side of the flower grows more than the other. Similarly, the expression of floral binding protein genes contributes to the positioning of floral organs symmetrically or asymmetrically. Cytoskeletal organization. Cytoskeletal organization. The plant cell cytoskeleton, which is composed of microtubules and actin filaments, also plays an essential role in establishing symmetry. The microtubules help in organizing the plane of division in the cell during early developmental stages, and their orientation determines how the cell divides and contributes to the overall symmetry of the structure. Cell expansion and growth patterns. Symmetry in plants is also influenced by how cells expand after division. In radial symmetry, cells expand uniformly, maintaining an even growth pattern. In bilateral symmetry, asymmetric cell expansion occurs, leading to differential growth along one plane of the structure, as seen in the zygomorphic flowers of leguminosae. Symmetry and developmental evolution in plants. The evolution of symmetry in plants plays a crucial role in their survival and reproduction. Radial symmetry often aids in efficient pollination because flowers with radial symmetry are often more accessible to a wide range of pollinators. Bilateral symmetry, on the other hand, has evolved in some plant species to specialize the flower for specific pollinators, leading to adaptive radiation and enhanced pollinator attraction. Previous year CSIR net questions. Symmetry in plants. CSIR net JRF December 2018. Which of the following types of symmetry is found in most flowers of the family Leguminosae, e.g., pea plants? Answer: bilateral symmetry. Explanation: flowers of the Leguminosae family exhibit bilateral symmetry, where the flower can only be divided into two mirror image halves along one plane. CSIR net JRF June 2020. What is the role of the CYCLOIDEA (CYC) gene in plants? Answer: it regulates floral symmetry in plants. Explanation: the cycloidea CYC gene is crucial for establishing bilateral symmetry in flowers, particularly in species like the pea plant. CSIR net JRF December 2017. In which stage of plant development does the establishment of radial symmetry typically occur? Answer: during the early stages of embryo development. Explanation: radial symmetry in plants, such as in flowers and roots, is established early in development, typically during the embryonic stages. This lecture on the establishment of symmetry in plants is designed to provide a detailed, comprehensive understanding of the topic, aligning with the CSIR net JRF Life Sciences exam standards. Let me know if you need further elaboration or the next topic. You said seed formation and germination. ChatGPT said seed formation and germination. CSIR net JRF Life Sciences lecture. Seed formation and germination are fundamental processes in plant reproduction and development. The formation of seeds begins after fertilization and plays a critical role in the life cycle of plants. It allows for the protection and dispersal of the plant's progeny, ensuring that the species can survive and thrive across diverse environments. Germination is the process by which a seed develops into a new plant, initiating its growth cycle. Both processes are regulated by complex physiological, biochemical, and environmental factors that ensure the seed's viability and successful establishment of the next generation. Seed formation. Seed formation begins after successful fertilization, which occurs when the male gamete sperm fuses with the female gamete egg within the ovule of the flower. The fertilized egg becomes a zygote, which develops into the embryo. Seed formation involves several key stages: embryo development, endosperm formation. Seed formation involves several key stages: embryo development, endosperm formation, and seed coat formation. Embryo development. The fertilized egg divides through mitosis, forming the early embryonic stages. The embryo develops into a mature structure that includes the cotyledons, seed leaves, epicotyl, the shoot above the cotyledons, hypocotyl, the region below the cotyledons, and the radicle, which will develop into the primary root. The embryo is essentially a miniature version of the adult plant, awaiting suitable environmental conditions for germination. Endosperm formation. In many plants, fertilization also results in the formation of the endosperm, a tissue that provides nourishment to the developing embryo. The endosperm is formed through a process called double fertilization in angiosperms, where one sperm cell fuses with the egg cell to form the zygote embryo, and the other sperm cell fuses with two polar nuclei to form the triploid endosperm. The endosperm stores starches, lipids, and proteins, which will be used during the seed's early stages of growth. Seed coat formation. The seed coat develops from the layers of the ovule. This protective layer surrounds the embryo and endosperm, providing mechanical protection against physical damage, desiccation, and pathogen invasion. The seed coat is typically tough and impermeable, preventing premature germination by blocking the entry of water and oxygen until conditions are favorable. The final seed structure consists of the embryo, endosperm, and seed coat. In some plants, such as eudicots, the seed's main food storage tissue is the endosperm, while in others like monocots, the endosperm may persist throughout the life of the seed. The seed's development concludes with seed maturation, during which the seed dehydrates, enters a state of dormancy, and is ready for dispersal. Seed dispersal. Once seeds are formed, they are dispersed from the parent plant. Dispersal mechanisms include wind, water, animals, and explosive mechanisms. Each plant species has evolved specific mechanisms to enhance its chances of spreading its seeds to new, suitable locations for growth. Wind dispersal. Seeds with lightweight structures or wing-like appendages, e.g., dandelion seeds, are carried by the wind over long distances. Animal dispersal. Many plants rely on animals to transport their seeds. Seeds may attach to animal fur or be ingested and later excreted, allowing them to be deposited in a new location. Water dispersal. Some seeds are adapted for flotation in water and are dispersed by rivers, streams, or ocean currents, e.g., coconut seeds. Explosive dispersal. Certain plants, like the violet, have mechanisms that cause the seed pod to burst open and throw the seeds away from the parent plant. Seed germination. Germination is the process by which a seed begins to grow and develop into a new plant. It is initiated when the seed absorbs water and resumes metabolic activity after a period of dormancy. Successful germination is dependent on various factors, including temperature, moisture, oxygen, and light. The stages of seed germination can be divided into several key phases: imbibition, activation, growth, and emergence. Imbibition. The first step in germination is the absorption of water, known as imbibition. Water enters the seed, causing it to swell. This rehydration triggers the activation of enzymes that break down stored nutrients in the seed, providing the energy needed for the growth of the embryo. Imbibition also softens the seed coat, allowing the radicle to emerge. Activation. After imbibition, metabolic processes within the seed become active. The stored food reserves start. The stored food reserves starch, proteins, and lipids are converted into usable energy and building blocks. Key enzymes like amylases to break down starch and proteases to break down proteins are activated. During this phase, cellular respiration resonates, providing energy for the growth of the embryo. Growth. As metabolic processes ramp up, the embryo starts to grow. The radicle is the first part of the embryo to emerge, establishing the plant's root system. The shoot apical follows, growing upward toward the soil surface. This growth is controlled by hormones such as gibberellins, which promote cell elongation and division in the embryo. Emergence. The final stage of germination is the emergence of the shoot above the soil surface. Once the seedling breaks through the soil, it begins to photosynthesize and establish itself as an independent plant. Factors affecting seed germination. Water availability. Seeds require an adequate amount of water for imbibition to activate their metabolic processes. Lack of water can prevent germination. Temperature. Each plant species has an optimal temperature range for germination. Extremes of temperature, either too hot or too cold, can inhibit or delay germination. Oxygen. Seeds need oxygen for cellular respiration during germination. Poor oxygen availability can limit the energy required for growth. Light. Some seeds require light to germinate, while others prefer darkness. Light-sensitive seeds have photoreceptors that influence the timing of germination. Dormancy and breaking dormancy. Seed dormancy is a natural state of inactivity or reduced metabolic activity in seeds, preventing germination under unfavorable conditions. Dormancy ensures that seeds do not germinate prematurely and ensures that they wait for the optimal environmental conditions. Physical dormancy is caused by a hard seed coat, while physiological dormancy is due to factors like the need for cold or light exposure. Dormancy can be broken by methods such as scarification, mechanically weakening the seed coat, or stratification, exposure to cold temperatures for a period. Previous year CSIR net questions. Seed formation and germination. CSI. Previous year CSIR net questions. Seed formation and germination. CSIR net JRF December 2019. What is the primary function of the endosperm in seeds? Answer. The primary function of the endosperm is to provide nutrients to the developing embryo. Explanation. The endosperm serves as a food reservoir for the developing embryo, providing essential nutrients during seed germination. CSIR net JRF June 2020. In which part of the seed does the embryo develop after fertilization? Answer. The embryo develops in the fertilized ovule. Explanation. After fertilization, the fertilized egg zygote divides to form the embryo, which is contained within the seed. CSIR net JRF December 2018. Which of the following conditions is not required for seed germination? A. Water. B. Light. C. Oxygen. D. Cold temperature. Answer. D. Cold temperature. Explanation. While some seeds require cold temperatures for dormancy breaking, most seeds do not require cold temperatures for germination. Water, oxygen, and light for some species are essential. This lecture on seed formation and germination covers essential concepts for the CSIR net JRF Life Sciences exam. Let me know if you'd like to proceed with another topic or require further elaboration. You said. Morphogenesis and organogenesis in animals. ChatGPT said. Morphogenesis and organogenesis in animals. CSIR net JRF Life Sciences lecture. Morphogenesis and organogenesis are crucial processes in animal development that determine the final form and function of an organism. These processes occur after fertilization and embryo development, ultimately resulting in the formation of complex structures and organ systems. Morphogenesis refers to the development of form and structure during the early stages of development, while organogenesis is the process by which specific organs and tissues are formed from the primary embryonic layers. Morphogenesis in animals. Morphogenesis is a biological process that shapes the body-plan organism. It involves the coordinated regulation of cell division, differentiation, migration, and death to generate the diverse structures in the organism. During morphogenesis, cells interact with each other in their surrounding environment, which drives changes in their shape, position, and function. Morphogenesis includes processes such as cell shape changes, cell migration, apoptosis, and formation of body axes. Cell shape changes. Cells undergo changes in shape to contribute to the overall structure of the developing organism. This includes apical basal polarity, where the cells develop distinct orientations that allow them to form tissues with a functional arrangement. For example, during gastrulation, cells at the surface of the embryo change shape to form specific regions such as the ectoderm, mesoderm, and endoderm. Cell migration. The movement of cells within the developing embryo is vital for morphogenesis. Cells can migrate to new locations during the development of various tissues and organs. A classic example of cell migration is during neurulation, where neural crest cells migrate to form structures like the peripheral nervous system. Apoptosis. Programmed cell death or apoptosis plays a crucial role in shaping organs and removing unwanted structures during morphogenesis. An example of apoptosis in morphogenesis is the formation of fingers and toes during limb development. The cells between developing digits undergo apoptosis, leaving behind the separated fingers. Formation of body axes. The establishment of the body axis, anterior, posterior, head, tail, dorsal, ventral, back, belly, and left, right axes, is an essential part of morphogenesis. These axes are established during the early stages of embryonic development and guide the spatial arrangement of cells. This is regulated by molecular signals like WNT, Notch, BMP, bone morphogenic proteins, and hedgehog signaling pathways. Organogenesis in animals. Organogenesis is the process by which the three germ layers, ectoderm, mesoderm, and endoderm, give rise to the various organs and tissues in the body. Organogenesis occurs after gastrulation when the basic body plan has been established. The process involves the differentiation of these germ layers into specific tissues and organs, a highly regulated process driven by genetic and signaling pathways. Ectoderm. The ectoderm forms the outermost layer of the embryo and gives rise to structures such as the nervous system, brain, spinal cord, and peripheral nerves, epidermis, skin, and sensory organs, eyes, ears. The differentiation of the ectoderm into these tissues is regulated by signaling molecules like BMPs and FGFs, fibroblast growth factors. An example of organogenesis from the ectoderm is neurulation, which involves the formation of the neural tube, the precursor to the brain and spinal cord. Mesoderm. The mesoderm lies between the ectoderm and endoderm and gives rise to structures such as the musculoskeletal system, bones, muscles, circulatory system, heart, blood vessels, and excretory system, kidneys. It also forms the gonads and connective tissues. Somites, which are derived from the mesoderm, play a critical role in the formation of the vertebral column and muscles. The notochord, which originates from the mesoderm, induces the formation of the neural tube from the ectoderm. Endoderm. The endoderm is the innermost germ layer, and it forms the lining of the digestive tract, respiratory system, and various glands, including the liver, pancreas, and thyroid gland. The endodermal cells differentiate into specialized cells that form the walls of these organs. For example, the gut tube forms from the endoderm and differentiates to form various parts of the digestive system, including the small and large intestines. Key processes in organogenesis induction. Organogenesis begins with the process of induction, where one group of cells influences the fate of another group of cells. Signaling molecules like morphogens, e.g., sonic hedgehog, shish BMPs, WNTs, are involved in this process. For instance, the Notch signaling pathway is crucial for the differentiation of cells into various cell types in the developing nervous system. Differentiation. As cells respond to induction signals, they undergo differentiation, where they become specialized into different types of cells, such as muscle cells, epithelial cells, or nerve cells. Differentiation is regulated by the activation and repression of specific genes in the genome. Patterning. Pattern formation during organogenesis is regulated by the coordinated action of morphogens that establish positional information within the developing tissue. This ensures that cells differentiate in the correct location and orientation, leading to the proper pattern of organs and tissues. Growth and remodeling. After the basic pattern of the organ is established, tissues and organs undergo growth and remodeling. For example, the formation of the heart involves the folding of the cardiac precursors into a tube, which then undergoes elongation, looping, and division to form the chambers of the heart. Similarly, the limbs undergo elongation and sculpting to form the digits. Examples of organogenesis: neurulation. Neurulation is one of the first stages of organogenesis, where the neural tube forms from the ectodermal tissue. The neural tube will later differentiate into the brain and spinal cord. This process is regulated by the Notch, WNT, and hedgehog signaling pathways. Limb development. Limb development is a complex process that involves signaling pathways such as FGFs, WNTs, and retinoic acid. During limb organogenesis, the limb buds form from the mesoderm and ectoderm, followed by differentiation of cells into muscles, bones, and skin. The zone of polarizing activity, ZPA, plays a key role in limb patterning by producing sonic hedgehog, shish, a morphogen that dictates the anterior-posterior axis of the limb. Heart development. The formation of the heart involves the fusion of two heart tubes, which then undergoes complex morphogenic movements such as looping and septation to form the four chambers of the heart. Signaling molecules such as BMPs, FGFs, and Notch are involved in the development of the heart. Previous year CSIR net questions. Morphogenesis and organogenesis. CSIR net JRF June 2020. Which signaling pathway is essential for the development of the neural tube during neurulation? Answer. The sonic hedgehog shish signaling pathway is essential for the development of the neural tube. Explanation. The shish signaling pathway regulates the patterning of the neural tube and the development of the central nervous system. CSIR net JRF December 2019. In animal development, which germ layer gives rise to the musculoskeletal system? Answer. The mesoderm gives rise to the musculoskeletal system, including muscles and bones. Explanation. The mesoderm is the middle germ layer responsible for forming the circulatory system, muscles, bones, and other connective tissues. CSIR net JRF June 2018. What is the role of apoptosis in morphogenesis? Answer. Apoptosis plays a role in shaping organs and removing unwanted cells or structures during morphogenesis. Explanation. Apoptosis eliminates cells that are no longer needed, such as those between the fingers during limb development, contributing to proper organ formation. This lecture on morphogenesis and organogenesis provides a detailed, exam-oriented overview of key processes in animal development. Let me know if you'd like to explore another topic or need further clarification. You said. Cell aggregation and differentiation in dictyostelium. ChatGPT said. Cell aggregation and differentiation in dictyostelium. CSIR net JRF Life Sciences lecture. Dictyostelium discoideum, commonly known as the slime mold, is a widely studied model organism in developmental biology, particularly for understanding the processes of cell aggregation and differentiation. This eukaryotic organism exhibits unique and Cell aggregation and differentiation in dictyostelium. CSIR net JRF Life Sciences lecture. Dictyostelium discoideum, commonly known as the slime mold, is a widely studied model organism in developmental biology, particularly for understanding the processes of cell aggregation and differentiation. This eukaryotic organism exhibits unique and dynamic cell behaviors, making it an excellent system for studying how cells communicate, aggregate, and differentiate in response to environmental signals. The study of dictyostelium has profound implications for understanding cell signaling, pattern formation, and tissue development. Cell aggregation in dictyostelium. In the life cycle of dictyostelium, individual amoeboid cells live as independent organisms that feed on bacteria. However, when food sources are depleted, the cells undergo a remarkable transition from a solitary existence to cooperative behavior, leading to cell aggregation in dictyostelium. In the life cycle of dictyostelium, individual amoeboid cells live as independent organisms that feed on bacteria. However, when food sources are depleted, the cells undergo a remarkable transition from a solitary existence to cooperative behavior, leading to the formation of a multicellular organism. This process is primarily governed by cell aggregation, which is initiated by external signaling. Aggregation signal, CAMP, cyclic adenosine monophosphate. When dictyostelium cells sense nutrient depletion, they begin secreting the signaling molecule CAMP in a rhythmic pattern. The cells move toward higher concentrations of CAMP, creating aggregation centers. This form of chemotaxis ensures that individual cells aggregate together at specific sites. The secretion of CAMP not only attracts cells but also acts as an autocrine signal, inducing further CAMP release and enhancing aggregation. Chemotaxis. Chemotaxis is the movement of cells in response to a chemical gradient. In dictyostelium, as cells secrete CAMP, they begin to migrate toward higher concentrations of this molecule. This leads to the aggregation of cells into large clusters, a process that is essential for the subsequent developmental stages. The actin cytoskeleton plays a crucial role in mediating this movement, where filamentous actin F-actin polymerizes at the front of the cell, and depolymerization occurs at the rear, allowing cell movement toward the CAMP gradient. Cell-cell communication. Aggregation also involves direct cell-to-cell communication. As cells begin to aggregate, they exchange signals through gap junctions, which allow the transfer of ions and molecules. These junctions help synchronize the behavior of the cells, ensuring that the aggregation process proceeds efficiently. Formation of the aggregation stream. The aggregation process culminates in the formation of visible aggregation streams, which are highly organized and directed collections of cells. These streams move toward a central point, where a pseudoplasmodium or slug will eventually form. The aggregation is a highly orchestrated event that involves the alignment of cells, their migration toward the aggregation center, and the initiation of further cellular processes that enable multicellular differentiation. Differentiation in dictyostelium. Aggregation also When dictyostelium cells sense nutrient depletion, they begin secreting the signaling molecule CAMP in a rhythmic pattern. The cells move toward higher concentrations of CAMP, creating aggregation centers. When dictyostelium cells sense nutrient depletion, they begin secreting the signaling molecule CAMP in a rhythmic pattern. The cells move toward higher concentrations of CAMP, creating aggregation centers. When dictyostelium cells sense nutrient depletion, they begin secreting the signaling molecule CAMP in a rhythmic pattern. The cells move toward higher concentrations of CAMP, creating aggregation centers. This form of chemotaxis ensures that individual cells aggregate together at specific sites. The cells move toward higher concentrations of CAMP, creating aggregation centers. This form of chemotaxis ensures that individual cells aggregate together at specific sites. The cells move toward higher concentrations of CAMP, creating aggregation centers. This form of chemotaxis ensures that individual cells aggregate together at specific sites. The secretion of CAMP not only attracts cells but also acts as an autocrine signal, inducing further CAMP release and enhancing aggregation. Chemotaxis. Chemotaxis is the movement of cells in response to a chemical gradient. In dictyostelium, as cells secrete CAMP, they begin to migrate toward higher concentrations of this molecule. This leads to the aggregation of cells into large clusters, a process that is essential for the subsequent developmental stages. The actin cytoskeleton plays a crucial role in mediating this movement, where filamentous actin F-actin polymerizes at the front of the cell, and depolymerization occurs at the rear, allowing cell movement toward the CAMP gradient. Cell-cell communication. Aggregation also involves direct cell-to-cell communication. As cells begin to aggregate, they exchange signals through gap junctions, which allow the transfer of ions and small molecules. These junctions help synchronize the behavior of the cells, ensuring that the aggregation process proceeds efficiently. Formation of the aggregation stream. The aggregation process culminates in the formation of visible aggregation streams, which are highly organized and directed collections of cells. These streams move toward a central point, where a pseudoplasmodium or slug will eventually form. The aggregation is a highly orchestrated event that involves the alignment of cells, their migration toward the aggregation center, and the initiation of further cellular processes that enable multicellular differentiation. Differentiation in endothelium. Once aggregation has occurred, the cells begin to differentiate into distinct cell types that will later form the structure of the multicellular organism. Dictyostelium exhibits two major differentiation outcomes: the formation of the slug and the development of spore cells. Formation of the slug. After aggregation, the cells form a slug-like structure that serves as the migratory stage of the organism. The slug is a highly organized, multicellular entity consisting of cells that are still capable of movement. This slug is a migratory form of dictyostelium that is capable of moving toward a more favorable environment, typically in search of new food sources. It is a transient structure that enables the organism to survive in an unfavorable environment, as it allows the cells to migrate together as a cohesive unit. Differentiation into fruiting body. Within the slug, cells undergo differentiation into different cell types. The two primary types of differentiated cells in dictyostelium are spore cells. A fraction of cells in the slug will differentiate into spore cells, which are highly resistant to environmental stress and capable of surviving in harsh conditions. These spores are the dormant form of the organism, which will germinate and reinitiate the life cycle when conditions become favorable. Stalk cells. The remaining cells differentiate into stalk cells, which will form the supporting structure that holds the spores. Stalk cells die after forming the stalk, and their primary role is to provide a physical structure for spore dispersal. Gene expression and transcriptional control. Differentiation in dictyostelium is tightly regulated at the genetic level. The CAMP signaling pathway continues to play a role during differentiation, particularly in the activation of specific genes that are involved in the formation of the slug and the fruiting body. Key transcription factors, such as GBF, growth factor binding protein DSPO1, and CAMP-dependent protein kinase PKA, regulate the expression of genes required for the differentiation of cells into spore or stalk types. This ensures that the differentiation process occurs in a coordinated manner, resulting in a functional multicellular organism. Role of signaling molecules. In addition to CAMP, other signaling molecules such as calcium ions, Ca2, and protein kinases contribute to the regulation of differentiation. CAMP induces the expression of genes that promote slug formation and guide the movement of cells during aggregation, while protein kinases are involved in regulating cellular responses to these signals, ensuring that differentiation occurs at the appropriate stage of development. Key stages in dictyostelium development: vegetative stage. The individual amoeboid cells feed on bacteria and multiply. During this phase, each cell lives independently, but they exhibit a primitive form of social behavior through signaling. Aggregation. Cells release CAMP in response to environmental cues, attracting nearby cells to aggregate together. This aggregation is a chemotactic process, culminates in the formation of an aggregation stream. Subformation. Once aggregated, the cells form a slug, which is a modal, multicellular structure. The cells within the slug are not yet differentiated into spore or stalk cells, but they move as a cohesive unit. Fruiting body formation. The slug eventually settles, and the cells differentiate into two main types: stalk cells, which form the support structure, and spore cells, which will become dormant and survive until conditions are favorable. Germination. When environmental conditions improve, the spore cells germinate and revert to the vegetative stage, completing the life cycle. Previous year CSIR net questions: cell aggregation and differentiation in dictyostelium, CSIR net JRF, June 2020. In dictyostelium, which molecule is responsible for initiating cell aggregation during? Answer: CAMP cyclic adenosine monophosphate is responsible for initiating cell aggregation during starvation. Explanation: CAMP serves as both an autocrine and paracrine signal, guiding cells toward aggregation centers during nutrient depletion. CSIR net JRF, December 2018. During the differentiation of dictyostelium cells, which of the following cell types contributes to the formation of the fruiting body stalk? Answer: stalk cells differentiate to form the stalk in the fruiting body. Explanation: stalk cells die after the differentiation, providing the structural support for the fruiting body. CSIR net JRF, June 2017. What is the primary role of the CAMP signal in the life cycle of dictyostelium? Answer: the primary role of CAMP is to mediate aggregation and coordinate the formation of multicellular structures. Explanation: CAMP acts as a signal for chemotaxis, guiding cells to aggregate and eventually form the slug and fruiting body. This lecture covers the key processes of cell aggregation and differentiation in dictyostelium, with an emphasis on the role of signaling molecules and the molecular basis of cell behavior. Let me know if you'd like to discuss another topic or need additional explanations. You said. Axis and formation in Drosophila. ChatGPT said. Axis and pattern formation in Drosophila. CSIR net JRF Life Sciences lecture. The study of pattern formation and axis specification in Drosophila melanogaster, a model organism in developmental biology, has provided fundamental insights into the mechanisms that govern the establishment of body plans during embryogenesis. The Drosophila embryo serves as an excellent model for understanding how complex patterns and structures are organized during development, and how the anterior, posterior AP, and dorsal ventral DV axes are established early in development. These processes are governed by morphogen gradients, transcription factors, and cell signaling pathways, all of which contribute to the spatial and temporal organization of the developing embryo. Establishment of axes in Drosophila. In Drosophila, the primary axes, anterior, posterior AP, dorsal ventral DV, and left, right LR, are established early during embryogenesis and are critical for proper body patterning and organogenesis. 1. Anterior, posterior AP axis formation. The anterior, posterior AP axis is established in the early stages of development through a combination of maternal and zygotic factors. The positioning of this axis determines the anterior head and posterior tail regions of the embryo. Maternal effect genes. The AP axis is initially set up by maternal effect genes, which are transcribed in the mother and deposited into the egg during oogenesis. These maternal mRNAs and proteins form gradients that provide spatial information for subsequent zygotic gene expression. Bicoid BCD and non-nose gradients. Bicoid BCD, a maternal transcription factor, is deposited at the anterior end of the embryo and forms a gradient with the highest concentration at the anterior and decreasing posteriorly. Bicoid promotes the expression of genes necessary for anterior structures, such as the head and thorax. Non-nose, another maternal protein, is localized at the posterior end of the embryo. Nanos acts in opposition to bicoid and inhibits the translation of the hunchback HB mRNA at the posterior, promoting posterior structure formation like the abdomen. Hunchback HB and caudal cow proteins. Hunchback is a target of bicoid, and its expression is activated at the anterior pole. Conversely, caudal is inhibited at the anterior and concentrated at the posterior pole. These gradients of bicoid and nanos establish a precise molecular framework that sets up the anterior-posterior organization. 2. Dorsal ventral DV axis formation. The dorsal ventral DV axis defines the back, dorsal, and belly ventral of the embryo. The establishment of this axis is crucial for the development of body structures like the gut, nervous system, and musculature. Dorsal gene. The dorsal protein is a key maternal transcription factor that plays a central role in the DV axis formation. Dorsal is initially uniformly distributed in the cytoplasm of the egg, but after fertilization, it undergoes relocation to the ventral side of the embryo, where it acts as a morphogen to pattern DX. Cytoplasmic localization. The gradient of dorsal protein is formed due to the polarized distribution of proteins such as toll and spatzel at the ventral surface of the embryo. Upon fertilization, toll receptors are activated by the ligand spatzel, leading to the activation of dorsal on the ventral side of the embryo. Dorsal protein function. The dorsal gradient is critical for determining the fates of different tissues along the DV axis. High concentrations of dorsal lead to the formation of mesodermal and ventral tissues, while lower concentrations allow the formation of ectodermal tissues. The dorsal gradient and zygotic genes. The dorsal gradient regulates the expression of key zygotic genes that define specific regions along the DV axis. Two. Dorsal ventral DV axis formation. The dorsal ventral DV axis defines the back, dorsal, and belly ventral of the embryo. The establishment of this axis is crucial for the development of body structures like the gut, nervous system, and musculature. Dorsal gene. The dorsal protein is a key maternal transcription factor that plays a central role in the DV axis formation. Dorsal is initially uniformly distributed in the cytoplasm of the egg, but after fertilization, it undergoes relocation to the ventral side of the embryo, where it acts as a morphogen to pattern the DV axis. Cytoplasmic localization. The gradient of dorsal protein is formed due to the polarized distribution of proteins such as toll and spatzel at the ventral surface of the embryo. Upon fertilization, toll receptors are activated by the ligand spatzel, leading to the activation of dorsal on the ventral side of the embryo. Dorsal protein function. The dorsal gradient is critical for determining the fates of different tissues along the DV axis. High concentrations of dorsal lead to the formation of mesodermal and ventral tissues, while lower concentrations allow the formation of ectodermal dorsal tissues. The dorsal gradient and zygotic genes. The dorsal gradient regulates the expression of key zygotic genes that define specific regions along the DV axis. For instance, high levels of dorsal activate genes like twist and snail, which are involved in mesodermal development, while lower levels activate genes like decapitoplegic DPP and SOG, which specify dorsal tissues. 3. Left-right LR axis formation. The left-right LR axis is crucial for the asymmetric positioning of organs in the embryo, such as the heart and gut. While not as thoroughly understood as the AT and DV axis, the establishment of this axis involves signaling pathways that generate left-right asymmetry in later developmental stages. In Drosophila, the left-right symmetry is influenced by signaling molecules like FGF, fibroblast growth factor, and Notch, which are involved in establishing organ asymmetries. Ciliary movement. Studies suggest that the movement of cilia in specific cells might play a role in the establishment of the LR axis by influencing the distribution of morphogens that drive asymmetry. Pattern formation in Drosophila. Pattern formation refers to the process by which cells within the embryo adopt distinct fates based on their position along the axes. This is governed by a series of morphogen gradients and transcriptional networks that coordinate the spatial expression of developmental genes. 1. Segmentation in Drosophila. Segmentation is a process by which the body of the embryo is divided into repeated segments, each of which will give rise to a segment of the adult body, such as segments of the thorax or abdomen. Gap genes. The first level of patterning occurs with the expression of gap genes, such as Krupple, Hunchback, and Knurps, which are regulated by the maternal bicoid and nanos gradients. These genes divide the embryo into broad regions and are essential for setting up more precise patterns. Pair rule genes. The second level of patterning is controlled by pair rule genes like even skipped and Fushi-Tarazu, which refine the segmental pattern into alternating segments. Segment polarity genes. The final level of segmentation involves segment polarity genes, such as wingless WG and engrailed N, which determine the anterior-posterior orientation within each segment. 2. Homeotic genes and Hox clusters. Once the basic body plan is established through segmentation, the differentiation of segment identity is further regulated by homeotic genes. These genes, especially the Hox genes, control the identity of individual segments along the AP axis. Hox genes. In Drosophila, the Hox genes are arranged in two clusters, the antennapedia and bithorax complexes, that are expressed in a specific order along the AP axis, corresponding to the position of each segment. For instance, antennapedia gene expression is required for the formation of thoracic segments, while bithorax genes help pattern the abdomen. Previous year CSIR net questions. Axis and pattern formation in Drosophila. CSIR net JRF June 2020. Which of the following maternal proteins is responsible for establishing the anterior-posterior axis in Drosophila? Answer. Bicoid BCD. Explanation. Bicoid is a maternal transcription factor that establishes the anterior-posterior axis by forming a gradient from the anterior to the posterior of the Drosophila embryo. CSIR net JRF December 2019. What is the role of the dorsal protein in Drosophila embryogenesis? Answer. The dorsal protein establishes the dorsal ventral axis and activates the transcription of genes like twist and snail in ventral regions. Explanation. Dorsal axis is a morphogen, with its gradient determining the fate of cells along the DV axis. CSIR net JRF June 2018. Which of the following gene products is involved in segmentation and responsible for dividing the embryo into broad regions along the AP axis? Answer. Gap genes. Explanation. Gap genes such as Krupple and Hunchback divide the embryo into broad regions during the initial stages of segmentation. Which of the following gene products is involved in segmentation and responsible for dividing the embryo into broad regions along the AP axis? Answer. Gap genes. Explanation. Gap genes such as Krupple and Hunchback divide the embryo into broad regions during the initial stages of segmentation. This lecture has provided a detailed understanding of axis formation and pattern formation in Drosophila, highlighting the molecular mechanisms that regulate the establishment of the body plan. The regulation by maternal effect genes, morphogen gradients, and the precise activation of segmentation and homeotic genes contributes to the orderly development of this model organism. Let me know if you'd like to dive deeper into any specific aspect. You said, Amphibia ChatGPT said. Developmental Biology of Amphibia, CSIR-NET JRF Life Sciences Lecture. Amphibians, such as frogs, salamanders, and caecilians, offer valuable insights into vertebrate embryology. Amphibians undergo a fascinating process of embryonic development, which includes early stages like fertilization, cleavage, gastrulation, and organogenesis. These organisms are particularly valuable for studying developmental processes because of their external fertilization, large eggs, and relatively simple body plans, which make them ideal models for experimental manipulation. Fertilization and early development in amphibia. The development of amphibians begins with external fertilization, which occurs in aquatic environments. For example, in frogs, the male releases sperm onto the eggs as the female lays them in water. 1. Fertilization. In amphibians, fertilization occurs externally in water. During fertilization, the sperm enters the egg, and the egg undergoes a series of biochemical and mechanical changes. The most significant change is the formation of the fertilization envelope that prevents polyspermy, ensuring that only one sperm fertilizes the egg. The sperm-egg interaction triggers a cascade of events that leads to the initiation of the zygote's development. The cortical granules in the egg release enzymes that alter the egg membrane, forming a barrier against additional sperm. 2. Cleavage. Following fertilization, the egg undergoes cleavage, which is a series of meiotic divisions that lead to the formation of smaller cells called blastomeres. In amphibians, the cleavage pattern is holoblastic complete, meaning that the entire egg divides. However, the cleavage is unequal in amphibians, resulting in smaller cells at the animal pole and larger cells at the vegetable pole. The cleavage in amphibians produces a blastula. In the frog and amphibian, this occurs after several rounds of division, and the blastula is formed with a central blastocele. The cells at the animal pole are smaller and more active, while the cells at the vegetable pole are larger and contain more yolk. 3. Gastrulation. Gastrulation is the next critical phase in amphibian development. During this phase, the three primary germ layers, ectoderm, mesoderm, and endoderm, are established. Invagination and involution. The process begins with invagination at the dorsal side of the embryo, forming a blastopore. As the blastopore deepens, it transforms into the archenteron primitive gut, and cells from the epibolic outer layer move inward. This process is similar to that seen in other vertebrates but has specific nuances in amphibians. The dorsal lip of the blastopore, also known as the sperm organizer, plays a crucial role in organizing the development of the nervous system and somites in the embryo. Formation of mesoderm. The mesoderm is induced by signals from the notochord and other signaling molecules, such as sonic hedgehog chiche, and forms structures such as the somites, segments of the mesoderm that give rise to the vertebrae and muscles. Formation of germ layers in amphibians. Ectoderm. The ectoderm gives rise to the nervous system, brain, spinal cord, skin, and sense organs. The neural tube formation is one of the first steps in nervous system development. Mesoderm. The mesoderm differentiates into several structures, including the musculoskeletal system, circulatory system, kidneys, and parts of the gonads. Endoderm. The endoderm gives rise to the gut, liver, and lungs in amphibians. Lungs are less developed than in other vertebrates. Neurulation and organogenesis in amphibians. Neurulation, the process by which the neural tube forms, is critical in vertebrate development. This process occurs shortly after gastrulation and involves the formation of the nervous system. 1. Neural tube formation. Primary neurulation. The process starts with the thickening of the ectoderm along the dorsal midline, which forms the neural plate. The lateral edges of the neural plate fold upwards, and the edges meet to form the neural tube. The neural tube eventually differentiates into the brain and spinal cord. The neural crest cells, which are derived from the ectoderm, migrate to form various tissues such as the peripheral nervous system, sensory ganglia, and parts of the heart. 2. Organogenesis. After neurulation, the process of organogenesis begins. The mesodermal derivatives, such as somites, further differentiate into tissues like muscles and vertebrae. The heart begins to form from the mesoderm as well, and the early stages of circulatory system development are seen in amphibians. The gut endodermal derivative begins to form as the archenteron develops into the stomach and intestines. Amphibian metamorphosis. One of the most remarkable features of amphibians is their ability to undergo metamorphosis. This process involves a radical transformation from the larval form to the adult form and is regulated by hormones like thyroxine. Larval stage. Most amphibians start life as aquatic larvae, e.g., tadpoles in frogs. During this stage, they have a tail and gills. Metamorphosis. As amphibians mature, they undergo metamorphosis, during which they lose their gills, develop lungs, and the tail resorts, transforming into the adult form capable of living on land. Previous year CSIR-NET questions. Amphibian development. CSIR-NET JRF June 2021. In amphibians, the dorsal lip of the blastopore plays a major role in which process? Answer. Organizer activity during gastrulation. Explanation. The dorsal lip of the blastopore in amphibians, known as the sperm organizer, plays a critical role in organizing the body plan by inducing the formation of the nervous system and other tissues. CSIR-NET JRF December 2019. Which of the following is a function of the notochord in amphibian development? Answer. Induces the formation of the neural tube. Explanation. The notochord in amphibians is essential for inducing the overlying ectoderm to form the neural tube, a key structure in nervous system development. CSIR-NET JRF June 2018. Which of the following statements about metamorphosis in amphibians is correct? Answer. Thyroid hormone regulates metamorphosis in amphibians. Explanation. Thyroxine, a thyroid hormone, is a key regulator of metamorphosis, leading to the transformation from larval to adult form in amphibians. This lecture has provided an in-depth overview of amphibian development, covering key stages from fertilization, cleavage, and gastrulation to organogenesis and metamorphosis. The amphibian developmental system continues to be a cornerstone in our understanding of vertebrate embryology. You said, Chick, ChatGPT said. Developmental biology of Chick, CSIR-NET JRF Life Sciences Lecture. The chick embryo has been one of the most extensively studied models in developmental biology due to its accessibility, rapid development, and well-defined stages. The study of chick embryology has significantly contributed to our understanding of vertebrate development, including key processes such as fertilization, gastrulation, organogenesis, and morphogenesis. The chick embryo is particularly valuable in studying embryonic development as it shares many developmental processes with other vertebrates, including humans. Here, we will explore the various stages of chick development, including early stages, gastrulation, neurulation, and organogenesis, along with its distinctive features. Fertilization and early development in chick 1. Fertilization. Fertilization in chicks occurs internally in the female reproductive tract. A mature ovum, which is an ova egg, is fertilized by sperm. The fertilized egg undergoes a series of meiotic divisions. Unlike many other vertebrates, chick eggs are telolecithal, meaning they have a large amount of yolk concentrated at the vegetable pole. This yolk provides nutrients for the developing embryo and leads to the formation of an unequal division during cleavage. 2. Cleavage. The chick undergoes discoidal cleavage, meaning the cleavage occurs in a disc-shaped area on the surface of the yolk, known as the blastodisc. The first few divisions result in a small number of cells in the embryo proper, while the rest of the yolk remains largely unaffected. The early cleavage is meroblastic partial, as only the blastodisc undergoes cleavage, while the yolk remains undivided. The blastodisc forms the blastoderm, which consists of two layers, the epiblast upper layer and the hypoblast lower layer. These layers play crucial roles in the formation of the germ layers during gastrulation. Gastrulation in chick development. Gastrulation is one of the most important stages of development, where the three primary germ layers, ectoderm, mesoderm, and endoderm, are formed. In chicks, gastrulation begins with the formation of the primitive streak, a thickened band of cells along the long axis of the embryo. 1. Formation of primitive streak. The primitive streak forms at the posterior end of the embryo and moves toward the anterior end. This structure is crucial because it marks the beginning of bilateral symmetry in the embryo and defines the future anterior-posterior axis. The cells of the epiblast migrate inward at the primitive streak, forming the mesoderm and endoderm layers. The remaining epiblast cells become the ectoderm. 2. Formation of germ layers. The cells that invaginate at the primitive streak form the mesoderm and endoderm, while the remaining epiblast forms the ectoderm. The mesoderm differentiates into structures like the somites, notochord, and blood vessels, while the endoderm gives rise to the gut and associated organs. The process of gastrulation is complete once the mesoderm is formed, and the notochord is fully established, signaling the onset of neurulation. Neurulation and organogenesis. After gastrulation, neurulation are next major step in chick development. This process leads to the formation of the neural tube, which will develop into the central nervous system (CNS). 1. Neural tube formation. Neurulation begins with the neural plate derived from the ectoderm forming a neural fold on the dorsal surface of the embryo. The neural folds move towards each other and fuse to form the neural tube, which will eventually give rise to the brain and spinal cord. The region of the neural tube near the posterior end will form the spinal cord, while the anterior portion forms the brain. 2. Somite formation. During the early stages of neurulation, the mesoderm differentiates into somites, which are paired blocks of mesodermal tissue located on either side of the neural tube. Somites give rise to structures such as the vertebrae, skeletal muscles, and dermis. 3. Organogenesis. As neurulation proceeds, the embryo enters the organogenesis phase, where various organ systems start to develop. Key events include the formation of the heart, liver, lungs, and kidneys from mesodermal and endodermal tissues. The circulatory system begins to form, and blood vessels start developing from mesodermal progenitors. The heart forms from mesodermal precursors and begins to function early in development, circulating nutrients to the growing tissues. Chick development beyond organogenesis. After organogenesis, the chick embryo undergoes a process called morphogenesis, where the organs continue to grow and refine their structure. Chick embryos are developed enough to be hatchlings after approximately 21 days of incubation, marking the end of embryonic development. Metamorphosis in chick post-embryonic development. Unlike amphibians, chicks do not undergo a significant metamorphic change, but their development from the embryo to the hatchling involves significant changes in morphology and physiology, such as the development of feathers, limbs, and the development of adult functions like respiration and feeding. Previous year CSIR net questions. Chick development CSIR net JRF June 2020. What is the function of the primitive streak in chick development? Answer: formation of the mesoderm and establishment of the body axis. Explanation: the primitive streak is the site where the mesoderm forms by the process of invagination, and it also defines the anterior-posterior axis of the embryo. CSIR net JRF December 2019. Which layer of the chick embryo gives rise to the neural tube? Answer: ectoderm. Explanation: the ectoderm gives rise to the neural tube during the process of neurulation in chick development. CSIR net JRF June 2018. Which of the following is the primary function of somites in chick development? Answer: formation of the vertebrae and skeletal muscles. Explanation: somites, derived from the mesoderm, give rise to the vertebrae, skeletal muscles, and dermis. This lecture provided a detailed analysis of chick development, covering key stages from fertilization, cleavage, and gastrulation to neurulation, organogenesis, and post-embryonic development. The chick embryo offers invaluable insights into vertebrate developmental processes and serves as a crucial model for understanding human embryology. You said, organogenesis. ChatGPT said. Organogenesis in chick invertebrates. CSIR net JRF Life Sciences lecture. Organogenesis is the process during which the three germ layers, ectoderm, mesoderm, and endoderm, differentiate into the organs and tissues of the body. It begins after gastrulation and proceeds through complex cellular interactions, signaling pathways, and morphogenic movements. Organogenesis is a critical stage in the development of all multicellular organisms, particularly invertebrates like the chick. During this period, the basic body plan is elaborated, and functional organs are formed, which are essential for the organism's survival post-birth or hatching. In chicks, this process occurs rapidly and leads to the formation of structures such as the heart, nervous system, lungs, kidneys, and digestive tract. In chicks, this process occurs rapidly and leads to the formation of structures such as the heart, nervous system, lungs, kidneys, and digestive tract. Stages and mechanisms of organogenesis in chick embryo 1. Formation of the heart. The heart is one of the first organs to form during chick development and is derived from mesodermal precursors. The heart begins to form at about day 2 of chick development, when the mesodermal cells in the anterior primitive streak differentiate to form the cardiogenic mesoderm. These cells migrate and condense to form the heart tube, which undergoes folding and looping to form the basic structure of the heart. Early heartbeats are observed around day 3, signaling the onset of circulatory functions. The heart tube eventually becomes subdivided into different chambers, atria and ventricles, which will later be further refined during post-embryonic development. 2. Development of the nervous system. The nervous system forms from the ectoderm, starting with the neural tube formed during neurulation. The neural tube differentiates into the brain and spinal cord. The anterior part of the neural tube develops into the brain, while the posterior part becomes the spinal cord. The brain forms 3 primary vesicles, prosencephalon, mesencephalon, and rhombencephalon, which later differentiate into more specific brain regions. The neural crest cells, which arise from the neural tube at the borders, migrate to various parts of the body and give rise to the peripheral nervous system and other structures like pigment cells, adrenal medulla, and craniofacial tissues. 3. Development of lungs. The lungs develop from the endoderm and mesoderm. During day 3 to 4 of chick development, the foregut anterior part of the digestive tract vaginates to form the lung buds. These buds grow and undergo branching morphogenesis to form the basic structures of the lungs. The surrounding mesoderm gives rise to bronchi and blood vessels that will be involved in gas exchange. The lung development continues after hatching, leading to functional lungs capable of oxygen exchange. 4. Formation of kidneys. The kidneys arise from the mesoderm, specifically from the intermediate mesoderm. The nephric duct forms early in development, and mesonephric kidneys, primitive kidneys, develop as an interim organ before the formation of metanephric kidneys, the functional adult kidneys. The process of kidney development involves the formation of structures such as the glomerulus, renal tubules, and ureters. Kidney function starts developing shortly after the formation of nephrons, but the full function is reached only after hatching. 5. Development of the digestive system. The digestive system begins to form early in chick development from the end. 4. Formation of kidneys. The kidneys arise from the mesoderm, specifically from the intermediate mesoderm. The nephric duct forms early in development, and mesonephric kidneys, primitive kidneys, develop as an interim organ before the formation of metanephric kidneys, the functional adult kidneys. The process of kidney development involves the formation of structures such as the glomerulus, renal tubules, and ureters. Kidney function starts developing shortly after the formation of nephrons, but the full function is reached only after hatching. 5. Development of the digestive system. The digestive system begins to form early in chick development from the endoderm, particularly the foregut. The foregut gives rise to structures like the esophagus, stomach, small intestine, and large intestine, while the hindgut forms the colon and rectum. The liver and pancreas are also derived from the endoderm of the gut. Development of digestive glands such as the salivary glands and the liver proceeds as the chick embryo grows. 6. Limb development. Limb development in chicks begins around day 3 when mesodermal cells at the lateral sides of the somites proliferate and condense to form the limb buds. The proximal distal axis of the limb is regulated by signaling molecules like FGF, fibroblast growth factors, which play a crucial role in limb elongation. The air apical ectodermal ridge at the tip of the limb bud regulates growth, and the ZPA zone of polarizing activity controls the anterior-posterior patterning. During later stages, the digits are formed, and chondrogenesis cartilage formation leads to the development of limb skeletons. Key regulatory mechanisms in organogenesis. Signaling pathways. Multiple signaling pathways control organ development. Some of the key pathways involved are WNT signaling, crucial for neural tube formation, heart formation, and limb development. Schist-sonic hedgehog, essential for limb patterning, nervous system development, and somite formation. FGF fibroblast growth factors, essential for cell proliferation and differentiation during limb and brain development. TGF beta, transforming growth factor beta, involved in mesodermal differentiation and cell differentiation in various tissues. Clinical relevance. Understanding the mechanisms of organogenesis in chicks has vast implications in medicine and genetics. It aids in the development of stem cell therapies, tissue engineering, and understanding congenital birth defects. Disorders in signaling pathways such as WNT and SHISH have been linked to malformations in the heart, neural tube, and limbs. Previous year CSIR net questions. Organogenesis CSIR net JRF June 2020. Which of the following organs in the chick embryo is derived from the mesoderm? Answer: heart. Explanation: the heart develops from mesodermal precursors during early embryonic development in chicks. CSIR net JRF December 2019. During organogenesis, the primary germ layer from which the lungs develop in chick embryos is answer: endoderm. Explanation: the lungs arise from the endoderm during the organogenesis phase. CSIR net JRF June 2018. Which structure gives rise to the kidneys during early chick embryogenesis? Answer: intermediate mesoderm. Explanation: the intermediate mesoderm is responsible for the development of the kidneys during organogenesis. This lecture provided an in-depth look at the process of organogenesis in chicks, highlighting the development of essential organs such as the heart, nervous system, lungs, kidneys, and digestive system. It also touched upon key signaling pathways and clinical relevance, crucial for understanding vertebrate development. The integration of these concepts will help reinforce your ability to tackle complex questions on organogenesis in exams. You said. Vulva formation in Caenorhabditis elegans. ChatGPT said. Vulva formation in Caenorhabditis elegans. CSIR net JRF Life Sciences lecture. The vulva formation in Caenorhabditis elegans, C. elegans, is one of the most studied examples of epithelial cell fate specification and organogenesis in developmental biology. This process has served as a model for understanding how cells receive positional cues to determine their fate during development. The formation of the vulva in C. elegans is essential for reproduction, and the mechanisms governing its development are highly conserved in other organisms, including humans. Overview of vulva formation in C. elegans. In C. elegans, the vulva is a structure that connects the uterus to the outside of the body, facilitating the passage of eggs during reproduction. Vulva formation involves a series of coordinated steps in which a set of vulval precursor cells, VPCs, are induced to differentiate and form the vulva. Key events in vulva formation: vulval precursor cells, VPCs. The vulva in C. elegans arises from 6 precursor cells located in the ventral hypodermis. These cells, known as VPCs, are located in a cluster around the midline of the animal's body and are numbered P3, P to P8, P. In the early stages of development, all 6 VPCs are equivalent in their potential, meaning they have the ability to become any of the different cell types that form the vulva. However, their fates are not fully determined at this stage. Inductive signaling by the anchor cell. The key to vulva formation is the interaction between the VPCs and the anchor cell, which is located in the gonadal arms of the hermaphrodite. The anchor cell secretes a signal that instructs the VPCs to differentiate. The signaling molecule involved in this process is LIN3, an epidermal growth factor EGF-like ligand, which is secreted by the anchor cell. The EGF receptor LET23 on the VPCs binds LIN3, activating a signaling cascade that influences the fate of the VPCs. Fate specification and induction. The LIN3 signaling pathway divides the VPCs into 3 different classes based on their proximity to the anchor cell. The nearest VPC, P6, P receives the strongest signal and becomes the primary vulval precursor. The 2 adjacent VPCs, P5, P and P7, P receive a moderate signal and become the secondary vulval precursors. The remaining VPCs, P3, P, P4, P, and P8, P receive little to no signal and remain in their undifferentiated state, eventually becoming hypodermal cells. The P6, P cell undergoes cell division to form the inner and outer cells of the vulva, which later develop into different parts of the vulval structure. Signaling pathways involved in fate determination. The LIN3 LET23 pathway activates several downstream signaling cascades, including the ROS-MEPK pathway, which leads to the activation of transcription factors that specify the vulval fate of the VPCs. Additionally, the Notch signaling pathway plays a crucial role in lateral inhibition between adjacent VPCs, ensuring that only the closest VPC, P6, P adopts the primary vulval precursor fate. This lateral inhibition prevents more than one cell from becoming the primary vulval precursor. The LIN12 and GLP1 genes are involved in regulating the Notch signaling pathway, promoting the differentiation of secondary precursors P5, P and P7, P while maintaining the hypodermal fate of other VPCs. Morphogenesis and vulval pore formation. Once the VPCs have differentiated, they undergo morphogenesis to form the vulval structure. The inner VPCs derived from P6, P form the inner layers of the vulva, while the outer VPCs form the outer layers. Cell-cell junctions and epithelial morphogenesis lead to the formation of the vulval pore. The P6, P derived cells elongate and fuse to form the vulva, which then connects to the uterus for egg passage. Role of genetic pathways. ROS-MEPK pathway. The LIN3 ligand activates the LET23 receptor on VPCs, leading to the activation of the ROS-MEPK pathway. This pathway regulates the transcription of genes that specify cell fate. Notch signaling. Lateral inhibition through Notch signaling ensures that only the closest VPCs, P6, P become the primary vulval precursor and prevents overproduction of vulval cells. WNT signaling. WNT signaling is involved in the positioning and alignment of the VPCs along the anterior posterior axis of the worm. It helps in ensuring proper morphogenesis of the vulva. GATA transcription factors. These transcription factors are essential for the differentiation of the VPCs into specific vulval tissues. The activation of GATA transcription factors like ELT2 and ELT3 is a critical step in the process. Experimental insights and importance. The study of vulva formation in C. elegans provides essential insights into how cells respond to positional signals and how these signals regulate cellular differentiation. The genetic pathways involved in vulva development are highly conserved across species, making C. elegans an important model organism for understanding developmental biology in higher organisms, including humans. Clinical relevance. Bulbar formation in C. elegans has been extensively studied in the context of organogenesis, cellular signaling, and cell fate specification. This research is valuable in understanding human developmental diseases, including birth defects and tumor formation, where similar signaling pathways might be disrupted. The EGF receptor LET23 and the ROS-MEPK pathway are conserved in humans and are frequently involved in cancer, particularly in epithelial cancers like breast cancer, where disruptions in the EGF signaling pathway can lead to uncontrolled cell proliferation. Previous year CSIR-NET questions. Bulbar formation in C. elegans, CSIR-NET JRF, June 2021. In Caenorhabditis elegans, which signaling pathway is primarily responsible for bulbar precursor cell fate specification? Answer: LIN3-LET23 EGF receptor pathway. Explanation: the LIN3 ligand from the anchor cell activates the LET23 EGF receptor on the BPCs, leading to bulbar precursor cell fate determination. CSIR-NET JRF, December 2020. Which gene product in Caenorhabditis elegans is responsible for the lateral inhibition that ensures only one bulbar precursor cell, P6, P becomes the primary precursor? Answer: LIN12. Explanation: LIN12, a Notch receptor, mediates lateral inhibition in BPCs, ensuring that only P6, P adopts the primary bulbar precursor fate. CSIR-NET JRF, June 2019. The development of vulva in Caenorhabditis elegans is influenced by the anchor cell secretion of which ligand? Answer: LIN3. Explanation: the LIN3 ligand, secreted by the anchor cell, activates the LET23 receptor on the bulbar precursor cells, leading to their differentiation. This lecture on bulbar formation in Caenorhabditis elegans emphasizes the intricate signaling pathways that govern organogenesis in this model organism. Understanding these processes is key to deciphering cellular fate decisions and morphogenesis in broader developmental contexts. You said, iLens induction. ChatGPT said. iLens induction, CSIR-NET JRF Life Sciences lecture. The induction of the iLens is a critical event in vertebrate eye development, which involves complex interactions between tissues, signaling pathways, and transcription factors. The formation of the iLens, a transparent structure responsible for focusing light onto the retina, is initiated by signals between the optic vesicle from the neural ectoderm and the surface ectoderm. Understanding the mechanisms underlying lens induction has important implications for developmental biology, stem cell research, and the study of eye diseases. Overview of iLens induction. iLens induction in vertebrates, including mammals, is initiated early during embryonic development. It begins with the formation of the optic vesicle, which extends from the forebrain. The optic vesicle interacts with the overlying surface ectoderm, inducing it to form the lens. The process of iLens induction is highly conserved across vertebrate species and involves a series of signaling events that ultimately lead to the differentiation of the lens placoid and its transformation into a functional lens. Key stages of lens induction: formation of the optic vesicle. The optic vesicle is an outgrowth of the diencephalon, part of the forebrain, and is one of the first signs of eye development. The optic vesicle invaginates and forms a structure that is closely opposed to the overlying surface ectoderm. The optic vesicle contains signals that initiate the induction of the lens from the surface ectoderm, starting the cascade of events that leads to the formation of the lens. Inductive interaction between optic vesicle and surface ectoderm. The induction of the lens is a result of reciprocal signaling between the optic vesicle and the surface ectoderm. The optic vesicle releases signaling molecules that activate specific transcription factors in the surface ectoderm, causing it to become competent to form the lens. Fibroblast growth factors, FGFs, bone morphogenic proteins, BMPs, and W. Overview of iLens induction. iLens induction in vertebrates, including mammals, is initiated early during embryonic development. It begins with the formation of the optic vesicle, which extends from the forebrain. The optic vesicle interacts with the overlying surface ectoderm, inducing it to form the lens. The process of iLens induction is highly conserved across vertebrate species and involves a series of signaling events that ultimately lead to the differentiation of the lens placoid and its transformation into a functional lens. Key stages of lens induction: formation of the optic vesicle. The optic vesicle is an outgrowth of the diencephalon, part of the forebrain, and is one of the first signs of eye development. The optic vesicle invaginates and forms a structure that is closely opposed to the overlying surface ectoderm. The optic vesicle contains signals that initiate induction of the lens from the surface ectoderm, starting the cascade of events that leads to the formation of the lens. Inductive interaction between optical and surface ectoderm. The induction of the lens is a result of reciprocal signaling between the optic vesicle and the surface ectoderm. The optic vesicle releases signaling molecules that activate specific transcription factors in the surface ectoderm, causing it to become competent to form the lens. Fibroblast growth factors, FGFs, bone morphogenic proteins, BMPs, and WNT signaling molecules are among the key molecules involved in this process. Formation of the lens placoid. The surface ectoderm, under the influence of signals from the optic vesicle, begins to thicken and form a lens placoid. The lens placoid is the first visible sign of the developing lens. This process is regulated by several key transcription factors, including PAX6 and QTFII, which are critical for lens placoid formation and early lens differentiation. PAX6 is a master regulator of eye development, and its expression in the surface ectoderm is essential for initiating lens formation. It activates downstream genes that specify the lens fate and maintain the identity of the developing lens. Lens formation and delamination. The lens placoid eventually invaginates and forms the lens vesicle. This is a crucial step in lens formation. The cells in the center of the lens vesicle differentiate into primary lens fibers, while those in the periphery remain as the lens epithelium. These primary lens fibers elongate and begin to express crystalline proteins, which are essential for the transparency and refractive properties of the mature lens. As the lens vesicle develops, the optic cup derived from the optic vesicle undergoes morphogenesis to form the retina, completing the structure of the eye. Reciprocal signaling. FGF signaling plays an essential role in both the induction of the lens and its subsequent differentiation. The optic vesicle secretes FGF8, which signals to the overlying ectoderm to form the lens placoid. In turn, the lens placoid releases signals that support the continued development of the optic vesicle, ensuring proper reciprocal signaling. Addition. Inductive interaction. The induction of the eye lens is a critical event in vertebrate eye development, which involves complex interactions between tissues, signaling pathways, and transcription factors. The formation of the eye lens, a transparent structure responsible for focusing light onto the retina, is initiated by signals between the optic vesicle from the neural ectoderm and the surface ectoderm. Understanding the mechanisms underlying lens induction has important implications for developmental biology, stem cell research, and the study of eye diseases. Overview of eye lens induction. Eye lens induction in vertebrates, including mammals, is initiated early during embryonic development. It begins with the formation of the optic vesicle, which extends from the forebrain. The optic vesicle interacts with the overlying surface ectoderm, inducing it to form the lens. The process of eye lens induction is highly conserved across vertebrate species and involves a series of signaling events that ultimately lead to the differentiation of the lens placoid and its transformation into a functional lens. Key stages of lens induction: formation of the optic vesicle. The optic vesicle is an outgrowth of the diencephalon, part of the forebrain, and is one of the first signs of eye development. The optic vesicle invaginates and forms a structure that is closely opposed to the overlying surface ectoderm. The optic vesicle contains signals that initiate the induction of the lens from the surface ectoderm, starting the cascade of events that leads to the formation of the lens. Inductive interaction between optic vesicle and surface ectoderm. The induction of the lens is a result of reciprocal signaling between the optic vesicle and the surface ectoderm. The optic vesicle releases signaling molecules that activate specific transcription factors in the surface ectoderm, causing it to become competent to form the lens. Fibroblast growth factors, FGFs, bone morphogenic proteins, BMPs, and WNT signaling molecules are among the key molecules involved in this process. Formation of the lens placoid. The surface ectoderm, under the influence of signals from the optic vesicle, begins to thicken and form a lens placoid. The lens placoid is the first visible sign of the developing lens. This process is regulated by several key transcription factors, including PAX6 and QTFII, which are critical for lens placoid formation and early lens differentiation. PAX6 is a master regulator of eye development, and its expression in the surface ectoderm is essential for initiating lens formation. It activates downstream genes that specify the lens fate and maintain the identity of the developing lens. Lens formation and delamination. The lens placoid eventually invaginates and forms the lens vesicle. This is a crucial step in lens formation. The cells in the center of the lens vesicle differentiate into primary lens fibers, while those in the periphery remain as the lens epithelium. These primary lens fibers elongate and begin to express crystalline proteins, which are essential for the transparency and refractive properties of the mature lens. As the lens vesicle develops, the optic cup derived from the optic vesicle undergoes morphogenesis to form the retina, completing the structure of the eye. Reciprocal signaling. FGF signaling plays an essential role in both the induction of the lens and its subsequent differentiation. The optic vesicle secretes FGF8, which signals to the overlying ectoderm to form the lens placoid. In turn, the lens placoid releases signals that support the continued development of the optic vesicle, ensuring proper reciprocal signaling. Additionally, BMP signaling from the optic vesicle helps in the suppression of other cell fates in the ectoderm, ensuring that the cells in the region of the lens placoid are committed to the lens fate. Notch signaling also plays a role in the regulation of cell fate decisions during lens development, preventing premature differentiation and maintaining the proper growth of the lens. Molecular players in lens induction. Several signaling pathways and molecules are key players in the induction of the lens. These include PAX6. As mentioned, PAX6 is essential for the initiation of lens formation. It is expressed in the surface ectoderm as a result of signals from the optic vesicle. PAX6 activates the expression of several genes that are critical for lens development. Fibroblast growth factors, FGFs. FGF8 from the optic vesicle is one of the critical signaling molecules involved in the induction of the lens placoid. FGF signaling also maintains lens development by activating downstream genes that regulate cell proliferation and differentiation. Bone morphogenic proteins, BMPs. BMP signaling from the optic vesicle helps to specify the fate of cells in the surface ectoderm, influencing the transition from a non-lens ectodermal fate to a lens-competent fate. WNT signaling. WNT signaling is involved in both the induction and maintenance of lens fate. It also plays a role in the formation of the optic cup and retinal development. Notch signaling. Notch signaling is important for lateral inhibition during the formation of the lens and retina, ensuring that the lens placoid forms at a specific position and that cells maintain their fates. Clinical relevance and applications. Understanding eye lens induction has broad implications for medical research. Disruptions in the signaling pathways involved in lens induction can lead to congenital cataracts, a leading cause of blindness. Understanding these pathways may help develop therapies for lens regeneration and cataract treatment. Additionally, knowledge of lens induction is relevant in the context of stem cell therapy, where scientists aim to regenerate eye tissues for patients suffering from degenerative eye diseases. Previous year CSIR net questions. Eye lens induction, CSIR net JRF December 2020. Which signaling pathway is crucial for the induction of the lens placoid during eye development in vertebrates? Answer: FGF, fibroblast growth factor signaling. Explanation: FGF8, secreted by the optic vesicle, is a key signaling molecule involved in the induction of the lens placoid from the surface ectoderm. CSIR net JRF June 2019. Which gene is considered the master regulator of lens development in vertebrates? Answer: PAX6. Explanation: PAX6 is the master regulator of eye and lens development. The induction of the eye lens is a critical event in vertebrate eye development, which involves complex interactions between tissues, signaling pathways, and transcription factors. The formation of the eye lens, a transparent structure responsible for focusing light onto the retina, is initiated by signals between the optic vesicle from the neural ectoderm and the surface ectoderm. Understanding the mechanisms underlying lens induction has important implications for developmental biology, stem cell research, and the study of eye diseases. Overview of eye lens induction. Eye lens induction in vertebrates, including mammals, is initiated early during embryonic development. It begins with the formation of the optic vesicle, which extends from the forebrain. The optic vesicle interacts with the overlying surface ectoderm, inducing it to form the lens. The process of eye lens induction is highly conserved across vertebrate species and involves a series of signaling events that ultimately lead to the differentiation of the lens placoid and its transformation into a functional lens. Key stages of lens formation of the optic vesicle. The optic vesicle is an outgrowth of the diencephalon, part of the forebrain, and is one of the first signs of eye development. The optic vesicle invaginates and forms a structure that is close. Key stages of lens induction formation of the optic vesicle. The optic vesicle is an outgrowth of the diencephalon, part of the forebrain, and is one of the first signs of eye development. The optic vesicle invaginates and forms a structure that is closely opposed to the overlying surface ectoderm. The optic vesicle is an outgrowth of the diencephalon, part of the forebrain, and is one of the first signs of eye development. The optic vesicle invaginates and forms a structure that is closely opposed to the overlying surface ectoderm. The optic vesicle contains signals that initiate the induction of the lens from the surface ectoderm, starting the cascade of events that leads to the formation of the lens. Inductive interaction between optic vesicle and surface ectoderm. The induction of the lens is a result of reciprocal signaling between the optic vesicle and the surface ectoderm. The optic vesicle releases signaling molecules that activate specific transcription factors in the surface ectoderm, causing it to become competent to form the lens. Fibroblast growth factors, FGFs, bone morphogenic proteins, BMPs, and WNT signaling molecules are among the key molecules involved in this process. Formation of the lens placoid. The surface ectoderm, under the influence of signals from the optic vesicle, begins to thicken and form a lens placoid. The lens placoid is the first visible sign of the developing lens. This process is regulated by several key transcription factors, including PAX6 and QTFII, which are critical for lens placoid formation and early lens differentiation. PAX6 is a master regulator of eye development, and its expression in the surface ectoderm is essential for initiating lens formation. It activates downstream genes that specify the lens fate and maintain the identity of the developing lens. Lens formation and delamination. The lens plaque eventually invaginates and forms the lens vesicle. This is a crucial step in lens formation. The cells in the center of the lens vesicle differentiate into primary lens fibers, while those in the periphery remain as the lens epithelium. These primary lens fibers elongate and begin to express crystalline proteins, which are essential for the transparency and refractive properties of the mature lens. As the lens vesicle develops, the optic cup derived from the optic vesicle undergoes morphogenesis to form the retina, completing the structure of the eye. Reciprocal signaling. FGF signaling plays an essential role in both the induction of the lens and its subsequent differentiation. The optic vesicle secretes FGF8, which signals to the overlying ectoderm to form the lens placode. In turn, the lens placode releases signals that support the continued development of the optic vesicle, ensuring proper reciprocal signaling. Additionally, BMP signaling from the optic vesicle helps in the suppression of other cell fates in the ectoderm, ensuring that the cells in the region of the lens placode are committed to the lens fate. Notch signaling also plays a role in the regulation of cell fate decisions during lens development, preventing premature differentiation and maintaining the proper growth of the lens. Molecular players in lens induction. Several signaling pathways and molecules are key players in the induction of the lens. These include PAX6. As mentioned, PAX6 is essential for the initiation of lens formation. It is expressed in the surface ectoderm as a result of signals from the optic vesicle. PAX6 activates the expression of several genes that are critical for lens development. Fibroblast growth factors FGF8 from the optic vesicle is one of the critical signaling molecules involved in the induction of the lens placode. FGF signaling also maintains lens development by activating downstream genes that regulate cell proliferation and differentiation. Bone morphogenic proteins BMP signaling from the optic vesicle helps to specify the fate of cells in the surface ectoderm, influencing the transition from a non-lens ectodermal fate to a lens-competent fate. WNT signaling is involved in both the induction and maintenance of lens fate. It also plays a role in the formation of the optic cup and retinal development. Notch signaling is important for lateral inhibition during the formation of the lens and retina, ensuring that the lens placode forms at a specific position and that cells mean their fates. Clinical relevance and applications. Understanding eye lens induction has broad implications for medical research. Disruptions in the signaling pathways involved in lens induction can lead to congenital cataracts, a leading cause of blindness. Understanding these pathways may help develop therapies for lens regeneration and cataract treatment. Additionally, knowledge of lens induction is relevant in the context of stem cell therapy. Understanding eye lens induction has broad implications for medical research. Disruptions in the signaling pathways involved in lens induction can lead to congenital cataracts, a leading cause of blindness. Understanding these pathways may help develop therapies for lens regeneration and cataract treatment. Additionally, knowledge of lens induction is relevant in the context of stem cell therapy, where scientists aim to regenerate eye tissues for patients suffering from degenerative eye diseases. Previous year CSIR net questions. Eye lens induction CSIR net JRF December 2020. Whittling pathway is crucial for the induction of the lens placode during eye development in vertebrates. Answer: FGF fibroblast growth factor signaling. Explanation: FGF8, secreted by the optic vesicle, is a key signaling molecule involved in the induction of the lens placode from the surface ectoderm. CSIR net JRF June 2019. Which gene is considered the master regulator of lens development in vertebrates? Answer: PAX6. Explanation: PAX6 is the master regulator of eye and lens development. Which signaling pathway is crucial for the induction of the lens placode during eye development in vertebrates? Answer: FGF fibroblast growth factor signaling. Explanation: FGF8, secreted by the optic vesicle, is a key signaling molecule involved in the induction of the lens placode from the surface ectoderm. CSIR net JRF June 2019. Which gene is considered the master regulator of lens development in vertebrates? Answer: PAX6. Explanation: PAX6 is the master regulator of eye and lens development. Its expression in the surface ectoderm is necessary for the initiation of lens formation. CSIR net JRF December 2018. What is the role of notch signaling in eye lens development? Answer: Notch signaling regulates the lateral inhibition necessary for lens placode formation, ensuring proper cell fate decisions during lens induction. Answer: Notch signaling regulates the lateral Previous year CSIR net questions. Eye lens induction CSIR net JRF December 2020. Which signaling pathway is crucial for the induction of the lens placode during eye development in vertebrates? Answer: FGF fibroblast growth factor signaling. Explanation: FGF8, secreted by the optic vesicle, is a key signaling molecule involved in the induction of the lens placode from the surface ectoderm. CSIR net JRF June 2019. Which gene is considered the master regulator of lens development in vertebrates? Answer: PAX6. Explanation: PAX6 is the master regulator of eye and lens development. Its expression in the surface ectoderm is necessary for the initiation of lens formation. CSIR net JRF December 2018. What is the role of notch signaling in eye lens development? Answer: Notch signaling regulates the lateral inhibition necessary for lens placode formation, ensuring proper cell fate decisions during lens induction. Explanation: Notch signaling prevents the formation of multiple lens placodes by inhibiting the differentiation of surrounding ectodermal cells. This lecture on eye lens induction highlights the complexity and precision required in the processes governing vertebrate eye development. Understanding these molecular mechanisms is crucial for advancing medical therapies related to eye diseases, regenerative medicine, and stem cell biology. You said. Limb development and regeneration in vertebrate. ChatGPT said. Limb development and regeneration in vertebrates. CSIR net JRF Life Sciences lecture. Limb development and regeneration in vertebrates are remarkable processes involving complex interactions between different tissues, signaling pathways, and genetic factors. The development of limbs from the embryonic stage to adulthood is crucial for the proper formation of functional appendages in vertebrates, including humans. Additionally, the ability of certain vertebrates, such as amphibians, to regenerate lost limbs presents a fascinating biological phenomenon that is still a subject of intense scientific investigation. Understanding these processes has significant implications for developmental biology, regenerative medicine, and the study of limb malformations and amputations. Limb development in vertebrates. The development of limbs in vertebrates is a highly regulated process that involves the formation of distinct tissues, such as muscle, bone, nerves, and blood vessels, which all contribute to the final structure and function of the limb. Limb development in vertebrates occurs in a series of well-defined stages and is controlled by several key signaling pathways, including FGF fibroblast growth factor, sonic hedgehog, and WNT signaling. This section explores the steps of limb development in detail. 1. Early limb bud formation. Limb development begins at an early embryonic stage, with the initiation of limb buds from the lateral plate mesoderm, which gives rise to the mesodermal progenitors of bones and muscles. The process begins with the formation of the limb field, regions of the embryonic body where limbs will develop. These fields are specified early in development and include cells that will give rise to the forelimbs and hind limbs. The signaling pathway that drives limb bud formation includes the FGF10 signaling from the lateral plate mesoderm, which activates the overlying ectoderm to form the apical ectodermal ridge hair. The air is essential for the elongation and proper development of the limb bud, as it secretes FGF8 and FGF4, which are crucial for the outgrowth of the limb. 2. Limb bud patterning.